Good afternoon. Thank you for joining us. I'm Pavita Howe, Director of Entrepreneurship Partnerships at the Rutgers Corporate Engagement Center, where we've created a new initiative to help bring visibility to the successes of Rutgers entrepreneurs and to help connect faculty, student, and alumni entrepreneurs to valuable resources and the broader Rutgers entrepreneurship community. We invite you to connect with us via our website and social channels, and we'll share that information at the end of this event. For today, I would like to welcome you to our first Rutgers Entrepreneurs event, co-sponsored by the Rutgers University Alumni Association. And special thanks goes to the Alumni Advancements events team for coordinating and managing this event. I am thrilled to welcome Dr. Andy Brooks, Chief Executive Officer and Chief Scientific Officer of Infinite Biologics, and Dr. Brooks, who formerly served as Chief Operating Officer and Director of Technology Development at Rutgers RUCDR Infinite Biologics, one of the world's largest academic biorepositories and a leader in biobanking, bioprocessing, and analytics. Infinite Biologics was launched after the sale of the RUCDR business and assets by Rutgers University in August of 2020. Andy, thank you so much for joining us today. It's a pleasure to have you.
Thanks for having me.
So it's wonderful to see you. We have several questions from our audience, but I wanted to start out by giving some history. Many of the people watching first heard about Infinite Biologics and RUCDR after COVID-19 hit and the saliva tests received emergency use authorization by the FDA. You were the first to receive an EUA, but Rutgers had existed for many, or RUCDR had existed for many years before that. So can you tell us about the evolution of RUCDR, how it came about, and what services you offered prior to COVID-19?
Sure. RUCDR was an acronym that was developed over 20 years ago by Jay Tishfield, who is the director of the Human Genetics Institute of New Jersey and was the founding department chairman for the Department of Genetics at Rutgers. And Jay coined the acronym for the Rutgers University Cell and DNA Repository. Over the years, we had rebranded into RUCDR Infinite Biologics because the work and the type of work that RUCDR had done at the outset as a very small academic program had grown into something much more. So when Jay started the RUCDR, before I even joined the faculty at Rutgers, it was developed to help make biological resources available for people that were looking for genetic variants in complex disorders. And the reason that this was so important was that there were not many samples available. So to be able to do genetic analysis that many years ago, it required very large sample numbers. It was very expensive. And in order to truly facilitate this kind of genetic-based research, it required that people share resources and people create resources. So Jay's vision and mission was to create the largest schizophrenia resource, for example, and he did, pulling in not only the NIH, but also just about every autism foundation and patient advocacy group to create a resource that's still the largest and most comprehensive in the world today. So building off of that, when I was recruited to join the team, it was to really expand the utility of facilitating research even further. And we took a mid-sized, manually based program, we automated it, we grew it. We then offered those same services to pharma and industry to build those collaborative relationships. And at that point in time, RUCDR, every 3 or 4 years, was doubling in size, doubling in revenue, until we got to a point as a next generation central laboratory, if you will, with a couple hundred employees, with $50 million or so in revenue, in a footprint of over 70,000 square feet on the Rutgers campus, that Dr. Barchi and Jay and myself had thought about how we get to the next level. And how we get to the next level was a function of, Rutgers doing what it does best, innovate, help programs grow, find things that are critical for our community, not just at Rutgers, but regionally, nationally, internationally, and then set them on a path to get to the next level. And that's what we did with the starting of Infinity Biologics.
Okay. And so what kinds of clients were you working with? Can you, give, I know some people had questions about some of the genetics services, Ancestry.com and 23andMe, and some of the companies you might have been working with.
Sure. So some naturally we can talk about, some we're not allowed to name based on our relationships. But what RUCDR and Infinity Biologics represents is a comprehensive biomaterial solution. So, and this is really how we segued into COVID in understanding biomaterials probably better than anyone else in the world, using all of our scientists, our expertise to maximize the utility of every sample. In order to do that, you have to develop technology and approaches to finding the best way to collect the sample, the best way to preserve it, the best way to process it, the best way to store it, and looking at a wide variety of ways in which you can analyze it, which is a very different approach than those that might have a clinical trial and say, oh, by the way, we collected this material, we would like to do this analysis, but we're not sure if we can do these other analyses. So we kind of turned the problem on its head, and that's how we operated as RUCDR and as we operate as Infinity Biologics. And through that approach, we've ended up working with most of the major pharma in the world, top 10 pharma, a large number of the top biotechs globally, in helping them develop a biomaterial plan specific for their applications. So we work across many other industries, such as direct-to-consumer genomics and genetics, mostly in the wellness and entertainment space, as precision medicine is starting to evolve into that area through physician-ordered, laboratory-developed tests, which are a little bit different than just understanding your genealogy, right, or the entertainment factor of some of the applications that companies like Helix used to support and things of that nature. And we also work among different industries. We work in the animal diagnostic space, doing inbred strain verification and outbred strain monitoring for large animal manufacturers, and have a number of programs that we built with commercial partners like Invigo and others to support those activities. And it's fascinating, the cross-fertilization between the NIH, so we're a very large NIH grant and contract contractor, grantee and contractor, through our existing and still growing relationship with Rutgers, helps make these marriages and these connections, which really, leads to a synergistic effect in discovery. And this is where the field that we're in is going. And I think, COVID has demonstrated that better than anything else, that the collaborative potential that we all know existed needs to be realized for all of us to live in better health. So.
Right. So so you were running all these services and then COVID came along. So tell us what happened.
Yeah, it did, kind of fall out of the sky. And one of the first things that we had looked at was there were some inquiries immediately, given that we are a CLIA lab in New Jersey, as to whether or not we knew anything about COVID testing. At that point, who knew anything really about COVID testing, other than, we were involved and still are with a number of companies such as Viral Clinics, which does a very large number of flu-based tests. They run the clinical trials that generates the data for the flu vaccine manufacturers. So, we sat down very, very quickly and gave some thought to what we were already learning were some of the limitations associated with testing. One, other reference labs had to step up. This had to be automated, and there had to be a better way to get people the test instead of test to the people, right? So if you think about a nasopharyngeal or a pharyngeal swab, it requires a lot of PPE for the healthcare professional. It requires them to be 6 inches away from the person that they're taking the test of, putting them at risk and potentially the person at risk. You know, there are a lot of people that needed to be tested and still need to be tested that are either not ambulatory, can't get to testing facilities, or just are scared, you know, to go outside with pre-existing medical conditions. So looking at saliva as the primary biomaterial for looking for COVID infection, we knew that that was possible based on other work we had done in the space with saliva. The question is, or at least for infectious disease, for respiratory viruses, the question was, could we identify quickly an approach, a device, a preservation agent that would do a number of things? One, you know, render the virus inactive, right? So we could work with it more safely and automate more easily in the lab. Two, before we knew about really the robustness of COVID-19, would it preserve the virus or would it get degraded on the way to the lab, and then we'd have different kinds of problems with testing? And you know, three, how robust was the collection? Because we've dealt with a lot of patients and have manufactured and assembled a lot of collection kits and working with viral clinics and know a lot about nasopharyngeal swabbing, how unpleasant it is, and how if the patient compliance is not high, we're not going to be able to combat the spread of infection. So all of those things combined led us to identifying some technologies we had worked with previously. And the way we managed the supply chain issue, which immediately, you know, broke the bank, if you will, was to partner with life sciences partners, piecing together a test and a technology that we knew would be relatively impervious to those kinds of supply chain. And knock on wood, it has been. We've had our challenges around standard laboratory reagents. Early on, it was, you can't get swabs, you know, things that you need to do for testing. You can't get media. Those are issues we never had. And then for certain reagents where large biotech companies were trying to scale up that had, or life sciences companies had not done this before, we chose two tremendous partners, PerkinElmer and Thermo Fisher, who were committed from day one to working with us, and not just solely with us after the fact. They're probably two of the largest providers of COVID-related reagents for nucleic acid work and for molecular testing. And we built a very robust pipeline, and the challenge from there was staffing and instrumentation and space. We've hired probably 600 or 700 people, if not more, in just a few months going back. Well, now it's not just a few months anymore. We've expanded our footprint for space tremendously. And quite honestly, this all started as we were still a part of Rutgers, but without Rutgers support and dedication to the COVID-19 pandemic, and not just for their faculty and their students and their staff, we would not have been able to make the impact that we have. Because as you know, Rutgers is now starting to think about how to come back to campus in a larger way. Now with the first doses of the vaccines being administered, we won't see anything in the way of, public vaccination to a level where it's protective, probably until April or May. But, Rutgers was very forward thinking, created a number of different opportunities for us to be successful and not just make an impact at the level of the state, which was recognized by Governor Murphy and still what we provide in capacity for all these state programs, but also, nationwide.
Right. And you raised a lot of questions. I had several, but for start, I know I had the privilege of working with you and your team during the early days when you were ramping up. And I know for a fact that your team was so dedicated, working around the clock, when many of us had the luxury, I guess you could call it luxury at that time, of being able to work from home and not have to worry about going to and from work and doing the things that needed to be done. You had people working around the clock And they were dealing with the same issues as the rest of us, you know, how to get their groceries, what to do about childcare when their kids were all home, and dealing with family issues and health issues. So how did you keep them motivated during that time?
So first of all, they're still working around the clock, 24/7, as testing has continued to be an extensive process. And to be honest, you know, during these times, it's very hard to keep people motivated. You know, we can't go and all go to a ball game. We can't, you know, have an outing and you know, grill outside and have everyone have their families and do a, you know, a carnival or anything that any one of the partners that we work with or our vendors would have loved to support and sponsored. And I think that just speaks to the unbelievable dedication of our team, both, you know, our full-time employees, the Rutgers employees that we work with, as well as our contract staff throughout the state and across all the laboratories that we support. We opened a lab in Minnesota about second week in December, probably about 2 months ago, and it was the you know, the same kinds of challenges. I think that our staff and everyone that's been involved has seen the higher purpose and the sense of commitment comes from the impact that they know they're making. And every opportunity that I get to talk about our lab and our team has always been to give the credit where credit is due. You know, a few of us had an idea. That's all it was. We went through the process of making sure that it would work. And everything from that moment on is really our staff, the Rutgers community, faculty, students, which came and volunteered, that still do so from time to time, and every single person every day that contributes in a tremendous way in making this happen.
That's wonderful. And I know they are still working hard. When I was in the lab, you were going from a few thousand to 10,000 to, you know, tens of thousands of tests. So what is capacity like at this time?
So with the instrumentation infrastructure we have and the space, we ultimately have the capacity now for about 150,000 samples a day. But not every day presents those kinds of sample challenges. And there are many different ways that samples get to us. For instance, the Department of Corrections in New Jersey tests everyone every other week, right? So they have lots of inmates and staff and guards that get tested every other week, and then their staff every week. Everyone has built a testing program that meets their unique needs, and there's not one testing program, you know, that suits all. The same thing around the holidays and people traveling. And I would say for the first time now, and I know it's going to come back to bite me just for saying it, is that we don't worry about capacity every second of every day anymore.
Right.
But we are far from the end of large scale testing, and things will ultimately transition to some really good point of care testing. You know, the differences between rapid testing and what we consider, you know, reference lab testing will always remain a difference, but it doesn't mean that that's not a great application for different kinds of screening, you know, scenarios. It really is a combination of different testing modalities that gives us the best coverage.
Right. And so there are a lot of questions about, you know, who has access to testing. I saw from some of the questions, I think some people didn't realize that testing has gone beyond the Rutgers community and even beyond New Jersey. You're testing in a lot of places. So people are asking how they can get access to testing, how widespread is saliva versus the other types of tests, and also what is turnaround time. Can you talk a little bit about that?
So I'll work backwards. The turnaround time for the test is 24 to 48 hours, 72 hours if a retest is needed due to an inconclusive result. the way that we built the lab and the automation that we have, we've always been at that turnaround time, which is very different than even now if you look at some of the pharmacies or city MDs of the world, where back in March and April, they would take a sample, but it would take 10 or 12 days to get a result. Now they won't take a sample until they know they have a slot to do the test. So the end result is the same in that it may take 5 or 7 days to get tested, or longer. And again, it's not like they're holding it. They do an outstanding job, the reference labs. We're all in this together. It's just, the sheer numbers and the way in which these tests are, presented. So that's our turnaround time. If you're interested to know just the test itself is only about 5 or 6 hours. I say only, if that's all you're doing for a small subset of samples. But you have to think about what it's like to open 50,000 Federal Express packages a day, literally. What it's like to decontaminate and work in a safe environment, the outside of those tubes. And then the setup of the automation for transfer, then everything's automated from there. There are some things that are just fixed time-wise, like PCR. It takes an hour for the protocol that we run. There are faster PCR protocols for point of care that are 45 minutes, and 15 minutes may sound like a lifetime, but then there are the reporting requirements, the data QC requirements. The last thing you want to do is knowingly get a test result wrong for anybody that runs and is a part of a clinical lab. So that's our turnaround time. As far as the efficacy of the test, our original study, and it's been borne out through larger sample numbers, and instead of throwing percentages around, because I think it leads to a lot of misinformation, the data that we have supports that a nasopharyngeal swab in a patient that is symptomatic or asymptomatic performs similarly to a saliva sample that's collected using at least the collection devices that we have validated. The Spectrum sDNA 1000 device was the one that we did in collaboration with Spectrum specifically on this application. Interestingly enough, if you look at the cases of false positives or false negatives, first you really have to understand them. And if there are follow-on questions, happy to go into that. But you'll find that there is pretty much for the data that we have, and in fact, this data was achievable only through some large clinical trials that were run through Rutgers, actually. Ray Panettiere, Jeff Carson, Marty Blazer were really the 3 PIs on the Healthy Coworker study, where they had the wherewithal to start to collect multiple sample types, saliva and nasopharyngeal swabs, before any shutdown, before knowing that healthcare professionals were the first ones exposed and have continued to follow them. So they truly had a source for people that were asymptomatic that then contracted COVID in the environment that they were in, mostly through work or whatever exposures. So all of that data adds up to show that it's just about 50-50. That if nasopharyngeal swab misses a positive result, that half the time the saliva will miss the positive result. But it's important to point out that we're talking about 1 to 2% of all results. You take that 1 to 2%, and it's about 50-50. And most of the times, it's at the level of detection of the assay, which is so close to the level of detection that even for the same collection twice, there's a chance that you may get a different result because the amount of virus that's being shedded is right at where we know the assay to be, its sensitivity to drop off.
Right.
Your last question, where do you go to get tested? If you're at home or your company or your business has a program in place, they usually provide information. We partner with a number of different telehealth partners like Vault Health, One Health IO, US Drug and Testing Centers. There are a number out there. And I think if you go online, you're not at a shortage for finding places that offer the test. And they also have to be very clear about the type of test they offer and show those labs that have emergency use authorizations versus those now that can run a test without that FDA authorization. That was a change 2 months ago, based on HHS's determination that the FDA doesn't necessarily have to regulate a high complexity molecular lab that could run this test. That was not the case up until that point in time. So there are a lot of options. If you're at Rutgers, I urge you to contact your departments, your programs that have information. Rutgers is expanding its testing program for staff and students this week, actually. And if you live in New Jersey, contact your county departments of health, contact the New Jersey Department of Health. We are now working across New Jersey in all of these areas to make testing accessible. And if you're unsuccessful with that, I'm happy to share some of our contact information at IBX, where we can get you to a testing source.
Okay, that's great. And I see there are a whole lot of questions coming in through chat. So for everybody that's asking, we may have to collect some of these questions, but we will try and get them to Dr. Brooks to try and get some answers afterwards as well and get back to people. I think we could probably talk for a long time and keep going with these questions. There was one that someone had asked about the Yale test. So Yale touted having a saliva test, and there were some some news articles saying they were first for certain things. So how does this test compare with Yale?
So the Yale test they're referring to is a test called Saliva Direct. And I haven't seen anything where they touted they were first. And to be honest, it really doesn't matter. They're at the time that we got our EUA, the very first one in March. No, in April, I'm sorry. There were 190 different, according to the FDA, saliva and or sputum-based applications. We were the first to receive an emergency use authorization. A month later, we were the first to receive for at-home collection, which really was the reason that we started this to begin with, to get the test to the patient. Saliva Direct, several months later, played off of the concept, knowing that saliva is, we all agree, a good source of biomaterials. They had published a paper back at the time we were doing our EUA to show that saliva was viable, but there was no test, right? It was just an academic exercise. And we had gone through a very similar exercise and realized that we needed to have a preservation agent. So what Saliva Direct is a mechanism that does not require a preservation agent. It also doesn't allow for at-home collection. You have to be near the lab where it's being collected. It's not as sensitive, but arguably, until we can determine how sensitive you need to say that you're infectious or not infectious, it does now have an emergency use authorization for a laboratory developed test. It got it, a little while ago. And their mission is to get testing into the hands of people that have molecular labs, instead of reinventing the wheel or struggling, to facilitate that. Whether that happens or not, depends on the lab. It depends on access to reagents. And their goal also was to hopefully provide a lower cost alternative. Although I think that, the cost associated with their evaluation was just based on the reagents and a manual lab process. It's a much bigger picture than that, right? And I think those that have looked into providing that soup to nuts solution, from physician orders to reporting, there's a lot of overhead associated with doing it. So, it's a saliva approach. We believe in saliva, so do they. We've collaborated on a number of things. We've been involved in a number of working groups to help professional sports teams and other things together. But I think that the biggest difference is we really feel that we need to mitigate risk by inactivating the virus when it's collected, in the event it's a positive sample, and having that added protection to preserve the nucleic acid to provide the most robust results, and going from there. But they're both qPCR assays, and both have made a significant impact in COVID testing.
Right. And there's certainly enough testing to go around.
Well, one would argue that there still is not. But there have been reports all along that there are labs that are capable, that are underutilized. There are lots of labs that are oversubscribed. So why are they still testing? It's a very complex landscape, and I can tell you, you can't underscore what companies like Vault have accomplished, or our partner Accurate Diagnostic Labs in New Jersey is the largest privately owned reference lab in dealing with the contractual nature, the regulatory nature, the collection, and then ultimately the reporting of those samples cannot be taken for granted. You have to operationalize this. It's easy for me in the lab to say, hey, I've got a process, I've got very cool robots, just line up the samples for me, and a couple hours later, here's your result. As we all know, and we're going to experience through the vaccination process, it's not that easy. And with the numbers that we're talking about, no one in my mind was prepared to be able to handle this.
Right. So I do want to talk about the vaccine, of course, too. But first, can you tell us, you transitioned from being part of Rutgers RUCDR to becoming CEO of a company. You have investors, you're still dealing with COVID, you've got employees. So what has that been like, and what has changed for you?
It's been exhausting.
I'm sure.
Lots and lots of long days. The asset purchase of RUCDR began long before COVID. As I mentioned, Jay and I talked with Dr. Barchi and Chris Malloy and the leadership at Rutgers about how can RUCDR continue to grow, benefit Rutgers, make Rutgers proud, but also get to the next level. And there shouldn't be an infrastructure in a public university to support a company that has aspirations that will generate hundreds of millions of dollars of revenue and solve big healthcare problems in a service-related setting. The real estate at Rutgers, the resources at Rutgers are to educating and creating the next 100 concepts. It'll be the next 100 RUCDRs. And that's how you make a huge impact as a university. So once we were agreed, which that was the easiest part of the whole thing, it was figuring out the operational and financial path forward, given the complexities of what I called our business really kind of reflected a turducken. So, a Super Bowl pastime. We were a business stuffed inside of a department, wrapped inside of an institute, stuffed inside of a university, right? How do you take that apart? And, it tastes so good, would you ever want to, right? But that's what we had to do. We had to pull that apart to make sure that, one, the Rutgers community was not left with any void whatsoever for the services that we provided. Two, that the consideration to all the jobs that have been created and maintained were all protected and in some instances transitioned such that we can continue to grow. And third, from a technology perspective and from an investment perspective, be able now to make the investments in the business that were required. This is not a fault of Rutgers. Again, Rutgers is a public university that has a very broad and at the same time focused mission, and growing companies is not one of them. The resources don't exist. That's why there's private equity, venture capital, angel investors, and other companies to buy other companies. So creating that opportunity was different than the way a traditional asset purchase agreement or acquisition happens, and you hire an investment banker, you put a data room together, and then you go with the highest bidder or the best offer. Our investor, stayed committed to this mission for a long time and made many concessions, I think, that otherwise would not be made in something like this, because they saw the vision and they believed in Rutgers and they believed in a Rutgers' role in getting this done. And it was just timing, a coincidence, if you will, The agreement was signed in the middle of June. It had been worked on a year before anyone knew what COVID was. And then we closed on August 17th and became Infinity Biologics.
Right. And there was a quick question about what revenues or benefits, financial benefits were going back to Rutgers as a result of this, creating the company and spinning out of Rutgers.
Yeah. So, I mean, Rutgers receives the overhead on any grants and contracts for work that Rutgers does. We plan on growing that portfolio. Rutgers now receives rent and payment for professional services as we still live among the Rutgers campus. We will grow outside of the Rutgers campus. It's the ongoing relationship in building and developing new technologies with Rutgers, and in fact now being a company that could license emerging technologies from Rutgers, which is a revenue stream or a potential revenue stream for any university. So, we work very hard on transitioning in the appropriate way, government grants and contracts, getting all the appropriate approvals, getting everything, checked out to make sure that, again, it goes without saying that anything was being done incorrectly, but to making sure that Rutgers receives and still receives the maximal academic benefit, and also the operational benefit of the services that are offered to the Rutgers community, which facilitates, a lot of research that may not be specifically what was RUCDR or IBX related. And I think, what Rutgers put together largely between Dr. Barchi and Chris Malloy and David Kimball created a program and a process that will continue to make Rutgers proud and let Rutgers benefit for many years to come.
That's great. And I'm sure people will be happy to hear that. So a question about the vaccine is here, it's being deployed. You recently announced services for vaccine storage and transport. So can you talk about, with the vaccine ready to go, what does that entail and what are you already doing to help deploy it?
Right. So the vaccine is here. It will be in growing numbers over the weeks to months. I still think the process and deployment is evolving from a public health policy and priority process. There's lots of good information on the NJDOH website about this and on the CDC website for those that want to educate themselves about the differences between vaccines, about what we know about their efficacy, and even right down to, if you're interested to learn how to handle them and transport them, something that when you do finally have the opportunity to go in and be vaccinated is interesting because you would never have thought about that, right? You walk in, they take out a vial from the refrigerator, they drop their syringe, they give you a shot in the arm and off you go. Typically, like for the flu vaccine or not, for a pandemic, obviously, you wouldn't give that much thought, right? When you take your child to the pediatrician's office and they get their vaccinations or their immunizations, you just don't think about it. So I think for those that have been following how this whole thing has played out, has been following some of the technology and want to understand what's important to you, for the first time, there's some really great resources. So I encourage you to go and look at them. Interestingly enough, part of what was RUCDR's core business, which is now IBX, is biosample storage and management. We have close to 20 million samples under management right now for research trials, cell lines, clinical trials, all different kinds of biomaterials that range from blood to stool to saliva to skin biopsies to tumors, all different kinds of things. And basically, if you can cut it out of you, scrape it off of you, spit it out, anything we could take off of you, or someone appropriately takes from inside of you, whether you deliver it yourself or it's taken from you from a surgeon, we know how to preserve it, process it, bank it, so that we can learn from it later on. And that goes back to how we started this conversation, which is what it was at the core of RUCDR, which has led to the birth of IBX and why we still continue to make an impact. And I think that the COVID-19 pandemic, for the most part, reinforces for me what we were built to do and what we can accomplish. And we will apply what we've learned from COVID, as well as our core principles and how we approach things, to lots of other, important problems. But once, we realized that cold chain storage was going to be critical for vaccination, we then again went back to the technologies that we use, the partners we have, leverage those relationships. As you can imagine, people just walking in off the street to Best Buy to buy a freezer, which is not the type of freezer you can use to store the Pfizer vaccine. And even if it was at the right temperature, is not something you would do for lots of reasons. Understanding what's required for backup power, not just, being on a different power grid, and not just having a small generator. We can power our entire facility through a series of generators that can be linked together. You have to have fuel plans, you have to have fuel trucks, you have to have the kinds of infrastructure people on staff that if a generator doesn't start. I'll give you an example. For Superstorm Sandy, which most of you on the phone, if you're from the Northeast, remember, certainly from New York and New Jersey and Pennsylvania and Connecticut, there were parts of our respective communities that were without power for weeks. RUCDR at the time was down for 7 seconds. We think it was 7 because the clock on the generator doesn't start counting until 10 seconds. We never lost power. We had mobile biorepositories that went into New York City and rescued the VA, NYU, and Bellevue. Over 100 years worth of research materials that otherwise would have been lost were brought back to New Jersey, triaged at our time. At that time, we had a partnership with Biostorage Technologies, and we were trucking frozen samples to Indianapolis, expanding into all of our storage facilities to literally rescue what every sample could otherwise be on a MasterCard commercial because it's priceless. We looked at that. We looked at how we enabled our network, how we look at the regulation over storage, all the temperature mapping requirements. So within a freezer, you may not know this, just because your freezer has a number, it doesn't mean it's the same temperature in every point in the freezer. You have to map that. The fact that data loggers, unlike just reading the number off of the outside of the freezer or a thermometer in the freezer, you need to record that every second of every day. So there's a temperature profile to see if there was a deviation, because if a freezer thawed and then froze, that vaccine may be compromised, but you would never know because you're not living there holding a little vial of vaccine to say, oh, boy, today was a warm day. And then the next day, you're frozen again. So taking all of that technology, what we do in cold chain logistics, made it again quite logical for us to participate in what we call, the last mile and facilitating the last inch. So as it gets from the manufacturer to UPS and FedEx to these hubs, with the help of McKesson providing the administration components, it needs to go to these regional and mostly privately owned, whether it's a hospital or what have you, storage hubs, and then get transported to the smaller units that are in the ward, that are in the hospital, and working hand in hand as we are, with local hospitals. We just had our first call with RWJ Barnabas that has done an outstanding job to prepare for the beginning, but now is thinking about how they support that constant stream. And certainly, of course, working with the Rutgers community to make sure that the vaccine is stored for access to those at Rutgers as a part of our VHS and the Rutgers community that will get vaccinated. So that is more part of our core business than even COVID ever was.
That's amazing. So, and actually it's something you just don't think about, but you also just put into perspective how stressed we all were about emptying out our own freezers and how trivial that was compared to what you were doing at the time. So that's great. So we don't have a lot of time left, but I don't know if everyone knows that you were in touch with a lot of very high profile people and organizations as a result of the testing and the visibility you got. You were testing for major league sports teams. You mentioned that you had actually done all the testing for the Biden administration. You were mentioned by the president for testing. And you told us about a call you got recently from someone that everybody knows. Can you tell us about some of those experiences?
Yeah, so there are a lot of things I will take away from this experience with the myriad of types of questions that people have. It's been actually fascinating. So even though we developed a test that we think has made a significant impact in helping people with the pandemic, the way in which people approach understanding components that are not necessarily part of our area of expertise is amazing. Because you do that, people may think that you're an expert in a lot of areas that you're not. And sadly, there's a lot of misinformation about COVID. It's gotten a lot better. In fact, Rutgers had, through Stephen Burley, a misinformation committee, which was very useful in helping people understand information about COVID. And many of you may have seen on Twitter, or at least read all the news stories about who's now the second wealthiest person in the world, Elon Musk, who took 4 rapid antigen tests and got 2 positives and 2 negative results in the same day. And as you know, Elon is polarizing when it comes to a lot of things, a brilliant individual. We've done testing for SpaceX since the beginning and have enabled their launches by testing not just the astronauts, but all of the people involved. So as you know, there was a launch that was coming up. Elon took these tests, didn't believe them. And to be honest, if I had an experience like his, I wouldn't have believed him either. It was 50-50, right? On four tests. So because of our relationship with SpaceX and being CEO, we tested Elon and provided a result that was consistent with what was then his symptoms and what he had reported. So I'm not at liberty to report, how he tested. You can read any number of news articles that are out there. He shared it with the world. And immediately after that test, we got a call from, from his camp, if you will, asking if he could talk with me and the chief medical officer from Vault, because he had questions about what the CDC had done and why is that important? What are some of the differences scientifically between rapid testing and non-rapid testing? How did this play into treatments? And really, you could almost, in talking with him, see his mind working about what kind of tech solutions could be brought to the data component of this. He asked about the thermodynamics of PCR and some things that were really fascinating in talking with him. Those are some of the experiences I will take away as being positive, and looking at, some very, very smart people and how they have assimilated this into their own personal life, but then I'm certain will make huge contributions back to the community. So anything that we can do and have done to facilitate that is, I take a lot of personal pride in that.
That's great. And you and your team have definitely been a source of pride for all of the Rutgers community. Other than having enough experiences to write a really great book, what do you see for you and the company on the other side of this pandemic?
I hadn't thought about writing a book, but now that's a pretty good idea. I've got notes and pictures and really, really some, I'm going to have to give you credit if this happens now, Pavita, but I don't know if you want to be a ghostwriter. You lived through this with us in the beginning. Really some fascinating experiences in, how people have reacted and have been humbled, and how some people that you thought might have the best interests in mind probably acted more selfishly out of fear. It's been amazing. I mean, what do I take away from this on the other side? We take away a bond we've built with our employees and our staff, and something that we will reflect on eternally, and how to build a better business to continue to make a significant impact in wellness and healthcare. You know, I take away with it not just a lot of pride, but a lot of knowledge around how we can make a different impact to lots of challenges we thought were insurmountable. And the reason we put them on the shelf was there were not hundreds of millions of people affected, or it wasn't, you know, as the president, when he, you know, reflected on what we've done as what we've accomplished is innovation under pressure. You know, pressure makes you think about a lot of things in a different way. I think that, you know, we now know how to address some of these things without having to have that critical pressure. And we're going to take this experience and really hopefully apply that to a lot of areas that we know, not just for COVID, but beyond COVID, that are going to help people in the future.
Right. Well, we were so happy to see you bringing people together and to have you work with other teams at Rutgers to make a difference during this time. So I think our time is just about up. There have been a lot of questions, and we will try and funnel some of those questions to you, and also help people connect to your team, as I see there are a lot of people asking to do so. But I'd just like to thank you so much for taking the time today. We really appreciate it. And it's been wonderful to hear more about the experience behind the scenes that most people didn't get to hear. So thank you so much.
Thank you, Pavita.
Thanks. And for all our audience, I'd like to thank you again for joining us today. Thanks to the Rutgers University Alumni Association for co-sponsoring and helping to organize this event. And also, I'd like to encourage everyone to connect with us. You can email us at entrepreneurs@research.rutgers.edu or go to our website, go.rutgers.edu/rutgerspreneurs, and also follow us on social media to hear more about Rutgers entrepreneurship. And we will post that information and make it available to you after the event as well. So thank you again. Thanks, Andy. And happy holidays to everyone.
Happy holidays. Bye-bye.